What are you guys asking about? The what? Where? Inside what? Cleavage furrow. None. Okay. This is a bioflix about cell respiration. At some point they will play. We hope. Either that or cause my computer to die in a miserable death. There it goes. Shh, quiet. There's hopes for it still. Is the sound on? You see, it's like playing but not playing. You see? It's very bizarre. Recent places. Come on. Of course it did. Perhaps. Quiet. Opening media. Sometime today. There. Last class we talked a little bit about ideas behind Lab 4. And Part E is still due. This weekend is the last day to do it. If you intend to do it, you probably need to do it today. Because if you don't do it today, the potatoes need to be done, stay over 24 hours over the water. You will collect data tomorrow. And that's the, if you want to put the report by Saturday, you would have to be able to do it. Today would be the last day you can do it. As far as investigation 13, part A was about enzyme rate. Now, I kind of already talked about this in the lecture, but what you notice is, of course, that the more concentration you have, the faster the rate you get. It doesn't mean much for the report, though, for those of you who actually turned it in. It doesn't mean much if you don't present that data visually, right? So if you should have done a graph where you graphed concentration, which we're changing the independent variable on the x-axis, and the dependent axis is the time, right? So if you did that, you will see that as you increase the concentration, the time gets faster and faster, and it should be exponentially so. Based on the class data that we got, even though we didn't explore a lot of data points, by the way, that's the other thing about doing experiments. If you have a lot of data points, you're going to bet a pattern. But it's still something like this, all right? So the more concentration you have, right, the less time it takes, right? And it's exponentially so. Of course, the other thing is that what do I do with the one that never went up? Well, you can think of it as infinity, right? So that data point should have been going towards infinity. Does that make sense to people? Yes? What's up? I can't stop to do that for you right now. Well, you can do it until Monday. I can't stop to set you up with that right now. I only have an hour of this class, and I'm teaching you about. And aren't you supposed to be somewhere?
No, I talked about this.
Did you tell me you were coming right now? Yesterday? Exactly, because I would have that out for you and stuff like that. I can't. It's locked right now. I'm sorry, but you can have it until Monday. You can do it after school as well, right? All right. So then, what was I saying? So the idea of Lab 13 is just exploring one factor that could affect the rate of photosynthesis. We also have enzyme catalysis. Now, of course, if you were graphing it the other way around, if you were graphing it, there's the virtual lab. If you do the virtual lab, it's even more important. What the virtual lab shows you is even more important. What they do is that they stop the reaction after a certain amount of time. And then in the virtual lab, it lets you quantify how much of the substrate there is left. So there's like a titration, like that you do in chemistry and stuff. And then when you get to the exact concentration of the substrate that's left, the color changes. And with that, you can actually tell. So then what happens is that if you let it run for a certain amount of time, you get this much. If you let it for a certain amount of time, you get this much. You know, and then with that, you actually get a pattern like this, which we talked about, right? So the idea is that at first the enzyme rate is very high, but then after a while it levels off, right? That's because if you increase, the enzyme starts working and destroying the substrate, but as less and less substrate becomes available, then you're at that point in the graph where it takes forever for the enzyme to do its job, right? And so the rate just basically flattens, right? And it can't go faster than that. This is the part of the graph that you're really interested in to see the rate at which the enzyme is helping out, right? Before you start getting the flat. The slope of this line there will be the rate of the enzymatic reaction, or the rate of the reaction when an enzyme is present, right? On part B, we're supposed to explore some other factor that could affect enzyme activity, such as a competitive inhibition. So you could have put other things other than what do you call substrate, and see if that would affect things. You could have put mess with the enzyme, change its temperature, or change the pH, and see what that was all about. Part C, which is due over the break, has to do with the researching the evolutionary significance and history of an enzyme of importance for biology, and making a video about it, where you actually collected this research from a variety of resources, and you teach us about that. You are supposed to post this video, right? And you know how I did a spreadsheet? I don't know if you know how I did a spreadsheet, but I sent on Sally a spreadsheet for the other one, the 6 genes activity, right? I'm going to send you tonight also a spreadsheet for this enzyme one. So what you're doing is you're putting the name of your enzyme, right? If you decide to do it, you should, because you have to do half the work, and a lot of you guys haven't done half the work. This is an opportunity to get those half the work. So you put the enzyme and the link to the video that you created, and then part D is actually part C is actually the peer review for lab 13, but part D is the research I'm talking about. And then you're supposed to also watch 3 videos from other students and do reflections on what you've learned from their videos. The same enzyme you use for that research should be used in lab 3, last part of lab 3, where you are doing a blast analysis of to answer some sort of question that you come up with that you could answer with blast. For example, it could be a question like, when did this enzyme evolve? You could answer that with blast, because you can see which branches of tree of life don't have it, right? You could answer it with blast, how important is this enzyme? Because if there was a highly conserved core structure that barely changed and is 99% the same in a variety of different branches, that would be evidence to show that it's very incredibly important and very slow mutating because of that. So in lab 3, there's a series of guiding questions that help you think of questions that you could ask and answer through blast about your enzyme, right? And that's still due during the break for that. The assignment about the 6 genes activity is due the day you come back for the break. There's lots of deadlines hitting during the break. Some of these deadlines will not be graded before the second 9 weeks ends, but especially with me going on trip and everything. But Whenever they get graded, they will count towards that quarter, whatever they get graded. So if I do have time to grade it for the second 9 weeks, they'll go for the second 9 weeks. If not, there'll be the grades that will start your third 9 weeks. Either way, stuff that you got to do. So look at that. Hopefully it will count for the second 9 weeks because some of you guys need the lab points to improve your lab grade. Remember that as soon as you come back from the break, also we have a test on Big Idea 2. The material that I'm about to cover is not part of Big Idea 2. All right? So that's that. We also worked on Investigation 5. Investigation 5 was about photosynthetic rate. The due date for that report, I forget what I don't have the calendar in front of me, but it should be pretty soon, right? Pretty soon you need to do the report for that. But before we can do the report, you guys have to post your average data on the lab data sheet. And a lot of you guys have not done that yet. Even those of you who had bad data. Lab 5 Part A actually worked. We have the setup, right? We got barely few to stand on the 30 centimeters from the light. And even less that standing on the farther than that. And then the light one actually went up, and then you put it in the dark, they stop going up, maybe even go down, right? That's what we want to see on that. So you have to be, and that the investigative lab actually tells you to do all sorts of analysis, including calculating a value they call E-value for the efficiency of the photosynthetic efficiency of the thing. And that is all part of what you need to do for Lab 5. But we can't do it if your groups to have data haven't collected the data. Otherwise, you're going to have to do it on your own individual data instead of the aggregate class data. So the idea is for you guys to post the data there. So I'm going to check the calendar right now to see when the Lab 5 part A, what you call report, was due. And there's also a part for Lab 5. We're going to do Part E soon. Part B of Lab 5 was about the stomata thing. Or is that stomata thing? Yes, where you're supposed to color it. Not color it, get an imprint of the stomata thing. If you use clear tape and did the imprint, you should have, first of all, it's kind of cool to actually see the stomata on the bottom of the leaf. You should notice patterns, like for example, some leaves have more than others. Some leaves, they look different from others. Some leaves have it more on top, more on bottom than they do on top. Some leaves don't even have anything on top, right? So it's interesting to look at that. But it's also interesting to find out why the leaves will have different amounts of stomata and show the variety of that. And if you look carefully, some parts of the leaves might have different amounts of stomata. And all of that stuff that's interesting to talk about. We already talked about the reflection about that in this class. Why would you only have stomata on top? Right? It's because if you have to be like a water plant or something like that stays on the bottom. Why would you have more stomata on the bottom than the top? Water conservation, remember that? So most plants are like that. Some have water on top and on the bottom. It probably means they live in very humid environments. They don't have to worry about water loss. Some don't even have stomata at all because they're trying to conserve water, or they live in water, so there's no need for stomata, right? So whatever those reasons may be. All right, so I'm actually looking here to find your lab report, Part 5. Let's see. Now, Part 5, we're going to do on, the report is due Monday? I don't think so. No, I'm talking about Part A through C. I'm talking about Part D, yes. Yes, we did. It was the one with the floating leaves, right? Part A was just observing the chloroplasts and noticing that they move around. And the idea there, of course, being that they're alive, right, inside the cell. And then there's the chloroplasts move around. They're not like this thing that just sits there waiting for the sunlight to hit it, right? The part in the Elodia, if you did the Elodia on Part 4, Lab 4, you get to see chloroplasts anyways, because that Elodia that's sitting in the tank, it's really easy to see chloroplasts in the Elodia. So those of you who haven't done that still, just get a slide, put it in the microscope, put a wet mount, put a slide on top, and look at the Elodia. You will see the chloroplasts in there. All right, of course that video that has it in suspension is kind of cool because they move around. The other thing, Part C, was about the pigments. Now the pigment thing is that you need to put a light beneath it so you can see them where they are. Some plants have more, some plants have less, but you should have seen the band where it started, and then you should have seen the other ones somewhere else, right? Now, each one travels less than the other does. The orange goes really far. Those of you who got an orange band, it goes really far, almost at like if the alcohol is here, the orange is right next to it, usually, right? That's the xanthophyll, whatever. Then you have some people got another yellow, another band up there, but then you always get a yellowish band, but you always get like a light green and a dark green band for the most part. And then usually the light green goes further than the dark green goes. Is that right? Right? And then you should be able to identify which pigments those are based on the RF value. Now, RF value just basically do the length or how much the distance that the pigment travel divided by the distance that the front travel. And then you can compare that. Now, remember that you're supposed to explain why did it go travel further. There's the one that has more affinity to the paper, that's stuck to the paper and travels slower than the other ones do, right? So the ones which are less, more soluble in the solvent and less affinity to the paper, they travel faster. And that's like xanthophyll's example of that. But the whole point of that is show that different leaves have different pigments and that the pigments themselves have different properties and different, just to see that there's lots of pigments in the plant, really. That's all it was about, right? And part E is about you inventing something. Now, some people already sent me a design and I said it was okay, but a lot of you haven't. Do not send me the design on Sunday night and expecting me to tell you if it's okay. All right? And remember, I told you last class how to have a good design. So just tell me, this is what we're going to do, Mr. Lee. We're going to change the temperature. That's not a design. What is your independent variable? Well, how are you going to measure? How are you going to change it? What is the dependent variable? How are you going to measure it? Safety protocols. Steps to increase precision. Steps to increase reliability. Right? Are you going to do multiple trials? How are you going to analyze the data? That kind of stuff. Well, for part D is like part E. You're just changing something. But there's a million things you can change. You can change the concentration of bicarbonate. I know somebody already has that. Correct? You could change the color of the light. I think somebody already has that. Correct? You can change the intensity of the light. Is what we already did. So you couldn't really do that again. You're not going to learn anything new. But you can change the type of the light. Maybe put a fluorescent light, a white light. If I'm saying, on the lab, investigative lab, there's a whole table full of the things that are part of what the experiment was, and you can change the way you do it. Anything you can change. You can change, somebody talked about changing the types of leaves. I think it was your group, right? So that's another thing. Is the photosynthetic efficient depending on the type of leaf? All of these things would be good, I think. So you just change a variable or change a part of the procedure and then see it, do your own investigation. You want to answer your own question. So, right, so then we're doing that on the last class. Still haven't seen. When did we do part D? What day?
Wednesday.
Was it Wednesday of last week?
It was a Thursday.
So that would mean that it was yesterday that was due, right? So I'm going to be nice because, you guys, the majority of you guys did not turn that in. Investigation 6 was done last Friday, though, all right? That means tomorrow investigation 6 right up is due. Now, remember I told you guys, although you don't have to, it's okay that it failed, right? For the most people, you can do the virtual lab, collect data from the virtual lab, and do a write-up on that, right? So the points, I'm not going to count against you the report points this time. I'm literally not putting in a total. But if you do it, you can get some points there. And God knows some of you guys need it. Regardless of that, everything other than the report is still fair game for Lab 6, including the extra tasks. And so when it says write-up due, that's the entire thing. So the reflection tasks, the extra tasks that are in there are due. So that's, due tomorrow. So if you, for example, if you don't do any of the extra or any of the reflection tasks, right, you're not going to be able to get those points, and then the total will go up when you don't go up. That's not a good thing to do. And if you're in a bad place in the investigation, you shouldn't do just half of it. You should do all of it, right? So you can catch up a little bit. And then the investigation part D is supposed to have been done yesterday, a week after we did it, right? But you guys haven't even put the lab data up on the table yet. So, it's okay if you give it to me by Saturday, all right? If you do a report by Saturday on what happened on part D, it's okay. But make sure to collaborate for the class so that we can actually have better data. So it's not just one trial, right? That you put your data online. Just transcribe your chart to that. Remember that there's a folder that says lab report writing with instructions on what you should do for a report. Tables, graphs, analysis that actually matches what it does, standard error of the mean, standard deviation. All that kind of stuff is better than not putting it. So when you get like a 1 out of 3 on the lab report, is because you did not follow the rubric. You didn't do what the rubric says. All right? So that's kind of the idea behind there. All right, so we talked about Lab 13 and Lab 5, kind of like a reflection on what it was all about. Your review books, who has a review book? Hand up high. You're supposed to have one. If you don't have one, buy one, okay? Your review books have a session that goes over the labs, right? Make sure as you complete each one, and we finish with each one, that you look through it and kind of process, what have I learned? What is this lab about? Because they ask questions about the lab. So even if you don't do it, and also remember those questions, the conceptual questions? This quarter we have had no time for FRQs. That is like the replacement of that. So not doing those is a failure to practice that. And yes, it's type. It's very easy to just grab somebody else's answers and post it on your report, right? But then, when the report is due, the final thing, because by the way, none of these labs have done the final report due except for Lab 6 tomorrow, which is just one part. That's why it's due tomorrow, all right? So when you should send your final one, I send it to a plagiarism scanner. And if you have the same answer as somebody else, you will get an F on that. By the way, when somebody cheats, I don't just give them a 0. I give them whatever you're supposed to get on that lab, negative on the grade book. I just warn you about that. Right? So don't copy from someone else. Don't submit something with a copy from someone else. There's too much to talk about in this topic that we have to hit today, which is cell division, which is not part of the next test. It's already I sent, by the way, the chapters which are due. And the 6 genes activities, because if you look at 3.A.2, which is what's actually supposed to happen today, but we are behind 1 day in the material. But if you look at 3.A.2, it says here in the bottom, certain human genetic disorders can be attributed to inheritance of a single gene traits or specific chromosomal changes, such as non-disjunction. You do have to have a general understanding of some of these disorders, right? And then, so this activity is designed for you two guys to look at what causes genetic disorders, right? By the way, mutations, disorder, it's very subjective depending on the environment, right? What you call disorder could be this next step of human evolution, right? Because bad is not necessarily, we talked about this already, right? Bad is necessarily bad. But if it is causing, we generally call disorder if it's causing us to die early or live a life of less well-being. So if you have to go to the hospital all the time because your cells keep clotting, because you have a cell that has a sickle shape that tends to clot around the junctions of the capillaries, and then you're going to have kinds of organ failure and trouble breathing, especially in higher places, you wouldn't be necessarily called adaptive. Of course, you can't get malaria though, so if malaria becomes a world pandemic, you'll probably be the only one to survive. And all of a sudden, it's not a bad thing. So it depends. Now, color blindness certainly doesn't seem like to be a good thing. By the way, there's a glass now, type of glasses, they're sunglasses looking, that actually fix color blindness. It's pretty awesome. All right? Now, Kleinfeld, so a lot of these things you need to know what they are. On my lecture series, I actually go over all of this in extent. I am really proud of my genetics lecture series, okay? Now, this topic is almost crazy to try to cover in a day because it's actually, if you look at it, it's about mitosis, meiosis, and Mendelian inheritance all put together. And so, the premise that we can cover in a day is ridiculous. But you notice that so let's do the basics, right? We can do at least some of it today, even on an early release day especially. So we're going to go further. We're definitely going to fall even further behind, but it's okay. We'll catch up later. By the way, on the third 9 weeks, we start coughing 2 or 3 EKs a day because they're different from these EKs. You'll see. All right? And by the way, all the reading will stop at the end of January, and then at the end of January, instead of reading assignments for the weekly suggested reading, you get a suggested review pace assignment. And I say suggested, but remember I said that you're supposed to do the suggested reading, and I know a lot of you guys haven't done it, and you will pay for it. I guarantee you. When you get your AP test grade back, and you will look like I should have read. That's what you're going to say. Trust me. And then when you're sitting on your freshman year in college, and you're taking Introduction to Biology instead of taking the first class that you're actually supposed to take for your major, and you came out, who's working towards an AA? Who's working towards an AA? All right? So you're working towards an AA and you get to college. Great. All the general requirements are fulfilled, right? Congratulations. And I'm so sorry. All right? So sorry. She's a Seminole, for future Seminole. And that's... Oh, good, good. I was worried about you there for a second. All right. No, but I mean, if that's the best one you get into, that's a good school, right? That's a... Oh, there you go. It's already decided. Why would you go to Tallahassee? All right, that's not... Oh, I'm just saying. Okay, I'm just kidding. Now, seriously, what I was saying is... I lost track of what I was saying. What was I saying?
That the Seminoles are off.
No, before that. Right, to keep up with the reading. If you have an AA and you have the general education requirements ready, when you go to your college, they'll throw you some other ones. They say, actually, these two things here don't really, Miami-Dade general education requirements are not the same as ours. But for the most part, you cover that. So maybe you have like 2 or 3 left to do to get what they consider their general education requirements. Because you already have an AA, right? That's great. So you took all these APs and Roman courses, you took care of general education requirements, you go to your college, in-state, if you go out of state, hopefully you have AP credits, but hopefully they take it. But if they don't, if you go in-state, you have all these AA credits, yay, I saved 2 years of my education, but you don't have introduction to biology and you want to be a biology major, or a pre-med, or a vet, or anything like that. The first thing you can do before you can do anything for your major is take this course. But since you only got a 3, and you actually got credit for biology, the one you're taking right now with Miami-Dade, which is not a biology for biology majors. It's or for majors of pre-med. It's just a general biology course, right? To fulfill a science requirement, which some of you guys are here for. If you get a 3, that's good enough. You fulfill a science requirement, done. But if you're doing this for a career of, say, medicine or microbiology or engineering or anything other reason, you're going to have to take real introduction to biology, which is a two-semester course with an attached lab for a total of 10 credits, right? And you can't take microbiology, anatomy, animal behavior, but genetics, biochemistry, anything that actually helps you graduate with your major before you take this course. Which means your first year of college, right, is going to be, you can't start your major if you're going to be a biology major. You understand what I'm saying? You can't start it. That means you do not save the year with the AA because that first year, all you do is introduction to biology. You with me, I'm saying? And then your second year, you can take the simplest classes. By the end of it, you're still there for 4 years. So the idea that the AA saves you time, it may be a myth, is what I'm trying to say. You know, it may be a myth. However, it does make your GPA go up. It make you look good. So for all those reasons, it's still worth doing it. But my point is that a 5 saves you time, saves you money in a different way. It actually gives you credits for what you need. And you're going to get a 3 and you'll be like, I should have read like Mr. Lima said I should have. All right? So, do the work. It's not too late. The hardest chapters are still ahead. Because the stuff that we've done so far is stuff that who have learned about these things, even maybe not in depth, but about these things in biology honors so far. Is that really something new of what we talked about? Maybe more depth, but not new. Correct? Are you with me, I'm saying? But the new stuff is coming. Read it. Study. Otherwise, you're not going to be good at it. For example, there's something new, I'm sure. That chapter 40, animal form and function, there's barely any biology honors classes that hit that stuff. Make sure you read chapter 40 before the test, because you're not going to be able to do it by intuition or by background knowledge kind of stuff. All right? That's going to be in the test, by the way. Back to this. All right? So, this is an onion root slide on Lab 7 next week. We're going to be looking at the first part of Lab 7. Is that, by the way, Part A of Lab 7 is one of those homework parts again. All right? Part A is about creating an animation for mitosis. All right? So, you literally grab clay and make chromosomes made out of clay. You know what I'm saying? And then you move them a little bit. And you move them a little bit. Are you with me? Stop motion animation of mitosis with you, with a voiceover by you, post it online. And you send it to me. Understand what I'm saying? You can Google it. There's a million of them made, so you can see how it's done. because this is a famous project that biologists just use. So it's on YouTube all over the place. And you can actually do meiosis as well. I don't know if it actually tells you to do meiosis as well. But you're just supposed to do that. You don't have to do it with clay. You can do it with coloring. But then you have to do like all of them and then do like a flip book. You know what I'm saying? Or you could do it with Legos and just have the Legos move around. Right? You can do whatever, whiteboard kind of thing. Right? So that, but it's whatever you do, that's what it is. And whatever the deadline of that lab is on the calendar, right? Part B though is a cool little kick little activity where we play a scavenge hunt. If you play honest biology with me, you've already done it. The scavenge hunt when you have to find the stages of mitosis. After I know that you know how to recognize the stages, then you go on a second activity where you actually count the number of stages on your root. There is an actual lab that they're supposed to do here, an investigative lab, where we actually put onion roots sitting inside of a chemical that makes them replicate faster. That's what it's all about. And then, of course, because of that, if you compare them with that onion root that was sitting in water, right, they should have more cells in mitosis. Does that make sense? Because it's replicating faster. We do not typically do that in the class, but if those of you who like to do it, it's a simple lab that actually works, unlike the other ones. And you can do it at the school, because all you need to do really is, we can do it. We can put the onions in the thing, but we're not going to have class time for you to do it. Because the class time this is why you need a class time. You need to cut the onion slide. You got to make the onion slide. And that's the hardest part. Right? It's a very important skill to learn how to make slides, but the tough part is cutting a slice thin enough for you to see something like this. And then you have to stain it. Right? The ones you're going to use are pre-made cut slides of onion root slides. Are you with me? But anyways, you can still do kind of an experiment with what we're going to do, and that's what I asked you to do. I asked you to scan different parts of the onion root to see if there's a difference in the growth rate. You would expect the part nearer to the tip to be growing faster than the part that's over here. Is there a difference between the amount of mitosis on the corners versus the mitosis in the center? So you can pick whichever one you want. Once you know how to identify them, you can actually then do that. And by the way, you can do a chi-square of that as well, because you can count the number of P, M, A, and T cells, all right, on the tip. And then you can do the same thing for the number of cells in the middle. And then you see a chi-square to see if there's a difference, right? So you prove with the data that there is a difference, especially if you do samples of multiple onion tips. And that's what part C is about. Instead, you can do edges versus middle. So this is a lab that's coming next week. Then we actually examine in the microscopes cancer cells, and we talked about what's the difference between cancer cells and why they're important and stuff like that. And then there's another kind of homework part, which you would do kind of like a paper on cancer and the history of cancer research and stuff like that. We are going to learn about this, the fact that surface area to volume ratio matters. So because surface area to volume ratio matters, and because, of course, life is all about replication, you got to divide, right? Because the cells can't keep growing forever. And so it gets to a point that the only way to keep growing is to split and grow again, right? So growth is always advantageous, but it gets to a point that you can't becomes unsustainable for the cell. So you grow in a different way. You grow by numbers, right? And of course, elephant doesn't have bigger cells. It just has more cells because of that limit to cell size, right? But the problems of eukaryotes is that eukaryotes have a complex, unlike cells, which are bacterial cells that just have DNA that can just be duplicated, sent to opposite sides, and then split in half. Eukaryotic cells can't do that because they have a nucleus. And so you need to evolve a process to separate the nucleus accurately so that you don't get a cancer. Because if you have erroneous separation of the nucleus, you don't really pass on your information correctly, correct? Right? And so mitosis must have evolved around the same time chromosomes evolved, right? In order to make sure that the nucleus was separated properly. So you can think of mitosis not as a cell division. In fact, it's not cell division. It's nuclear division, right? It's called karyokinesis. Or karyo means the DNA, the nucleus. And kinesis means separating that. Does that make sense? All right? So then that's what it's all about. chromosomes of these eukaryotes that do this kind of magic stuff, all right, this wonderful mitosis stuff, are linear. And that's another thing we have to talk about in this topic. Why are chromosomes around? What's the advantage of having chromosomes? If you didn't have chromosomes, you would have only... chromosomes exist so that you can actually have two copies of the information still attached into the moment it's ready for them to split. It makes the split process easier. The chromosome of bacteria, it's okay being just a single loop like that, but since we have thousands of times the DNA of bacteria, it's like trying to move a whole house without putting in boxes. It becomes too complicated. You're certainly going to make mistakes. You're going to break that thing trying to move it, right? So because our DNA is too long, we compartmentalize the DNA in small little pieces, right, which allows us to transfer them more accurately. That's the reason number one. So as soon as my eukaryotes started getting more complex with more DNA, right, we needed to do this process of having chromosomes. But because they're linear, there's a problem with the ends. We'll talk about that when we do DNA replication. But because they're linear, there's also cool things that come from that. The ability to pair them up so that you have two copies of each gene allows you to increase variety, right? Genes can now show up in multiple chromosomes, the same gene in multiple chromosomes, which allow for multiple sites of DNA activation. And even if you have a chromosome with a problem, maybe another one has the gene you want, all right? So it becomes a library that's more complex, allows you to have more variety. So it's like the equivalent of having multiple file folders instead of one file folder with all your files inside of it. Does that make sense? Most people put things in folders because it just becomes easier. And you could have your same file in different folders, with different versions of the file in different folders, right? But having things in folders makes it organized, better, copying, easier. So it's easier to draw a computer analogy here and understand what biology is. See, whenever there's a convergent of function, it's because there's a convergence in the need, correct? So you can even say there's an analogous structure between the folder structures of a computer, right, and the folder structures of a DNA inside of a cell. Does that make sense? That convergence is not accidental. It's because it is advantageous in both cases, right? Now, the thing about the cell cycle that everybody knows about, right, is that there's different stages, right? So as soon as a cell divides, it grows because it's a bit tinier as soon as it divides. And then it goes through a stage of DNA copy or synthesis, where you could, throughout this entire part, the cell is doing what it does, which is protein synthesis. And then goes a little bit more, prepares for division, and then it actually divides, right? Now, if you actually see muscle cells and nerve cells, because of what they are and the reason that they're always working, like cardiac muscle cells and brain cells, they can't divide. So you don't typically have brain cancer on neurons, because neurons don't split. So that's not going to be a thing that's typically going to happen. Brain cancer is typically cancer from somewhere else that ends up metastasizes to the brain, if that makes any sense. Or glial cells, like the one I had, which is the support cells that build the network or support the melanin sheath and stuff like that. And those glial cells are dividing very fast, and so they could be targets for cancer. But typically, neuron cancers are very rare because they don't split. No, you don't get muscle cancer very often because it doesn't split. It just gets larger. The cells stretch, but they don't really split much. After you're done, by the time you finish adolescence, you're not going to build. Well, you still split a few muscle cells, but your cardiac muscle, right, after it stops growing, so it's hard for them to split. They split a little bit, but very little. But neuron cells, after you stop building the brain, it doesn't split anymore. So actually, this is an interesting thing. Can the muscle still grow? Can the brain still grow? It can, so long as it has stem cells. So if you have like stem cells are cells which don't have specialized functions yet, right? So then you have lots of types of neurons, right, coming from that neuron stem cell. So how could your brain, which it is right now, still grow? You just said it doesn't divide, so how could that happen? Because it's not really these neurons, the end products, which are dividing, right? Your brain is growing from stem cells. Does that make sense? So the brain is not growing because a neuron splits into two neurons. Each neuron you're getting is coming from a neuronic stem cell. Does that make sense? Which is why if you find a way to stimulate neuronic stem cell replication among people who are losing neurons or have neuron problems like Parkinson's or other things like that, it could be a promise for them. right? So that they can actually improve that, even if it requires a transfer of stem cells from someone else, or getting stem cells from something other part of the body, which are, because see, neuronic stem cells come from other types of stem cells, which are even less specialized. Does that make sense? And so if I find this one somewhere else in the body, maybe I can induce that one to become a neuronic stem cell, and from that to become a neuron. But the mechanisms that make this happen are still unknown, and we don't know enough about them to actually tell the cells how to be the neuron yet. But the day we figure it out, we will be able to actually correct those kinds of problems, even in our brains, which are decaying. Right? Although I think it's the wrong route to go, in my opinion, because I think we'll transcend this biological machine someday. Right? By the way, calling a body a biological machine is counterintuitive, because the body is not a machine. A machine is something designed. We weren't designed, right? Body is a biological system. When you say something is like a machine, you almost intend a designer of a machine for a specific purpose. Life has no purpose, and it also wasn't designed. So you don't. So it's not. We're not machines, right? So that's interesting. That's an interesting thought. All right. So again, we already talked about chromosomes. Now notice, of course, that an actual chromosome is only visible right before they split up on the prophase. Although you can induce DNA to assume this shape with the right chemical stimulation. Now, the sister chromatids separate by breaking the centromere that connects them, and then they go to their opposite ends doing the division. This is the entire stages of mitosis. There are animations on the videos and animations folder. It is very important. Biology, bioflicks, animations in biology, and the Campbell biology ones are excellent ones. Take the time. It's worth it. When you're in the car, you're doing nothing, go to the videos and tutorials folder. Watch the animations in biology. Watch bioflicks. It will pop into your head. I get it. I see it. Mitosis makes sense. They have an awesome animation about the stages of the control of the cell cycle. They have an awesome animation about cells actually dividing. And I'm having trouble playing them, but you have it there. There's one by Campbell that I really wanted to show you, which actually shows you the myosin fibers that prepare the thing. You can actually see the DNA splitting. All of this is really awesome. So I'll put it on my archive of things to show you someday, right? But it's definitely something that you want to look at. So anyways, it goes beyond the scope of the course to know the actual stages of mitosis, believe it or not. I still teach them, of course. But the reason it's actually not important to think about stages is because there's no stages. We're the ones that call this a stage. But this is a whole process that happens seamlessly. Right? If I show you the video, you will see that there is no particular that's prophase. It's really hard to separate where it's happening. However, the DNA will start to coil up. I'm not going to sit here and explain to you guys how cells divide, right? Because you can read about that. But with the nucleus, you see the patterns, the nuclear envelope going away, right, and eventually returning at the very end. You see the DNA coiling, right, forming chromosomes, lining up in the middle, separating into sister chromatids, and then uncoiling, right? For the centrioles, if you're an animal cell, they will start building at the same time other things are happening, they'll start building the fibers, which eventually extend throughout the entire cell from pole to pole. The fibers will eventually attach to the centromeres, and then they will wiggle them back and forth. By the way, it's a misconception that the fibers pull them. That's not how it happens. In fact, it's from the other way around. The fibers shorten, depolymerize. Understand? So it's not like the centromere is pulling them, right? Even though there are aster fibers on this side holding the centromeres in place, and even though they kind of exert a kind of tension, the chromatids actually move by depolymerization of the fibers, of the tubulin fibers that make this thing. Now this thing, they go to opposite ends. So you see those patterns there. You can see it here on an animal cell, how it kind of looks like. You see the center anaphase, they're all lined up in the middle. So this is the scavenge hunt. That's what you have to try to find. In a plant cell, you actually see, this is telophase. You see typical telophase. You see the bundles and a hint of a little line, a cell wall building. On a plant cell, because the cell walls are rigid, they can't fold inwards like that, and so the cell wall is built from the inside out. Meanwhile, in an animal cell, because it's not rigid, it pinches inwards and separates in the cleavage furrow. Right? So, this is again showing you prophase, and that's what you're trying to look for. Or deorganization. So prophase looks like a deorganized nucleus. The spaghetti, the grayness of interphase is gone, and instead you get a darker. See, interphase, you don't have the darker spots. It's just grayish, right? By the time you get to prophase, it's going to be darker. So that's typical prophase. Or even more advanced prophase is when they're already kind of coalesce and separate into different sides. This is just talking about what I just talked about, the cleavage furrow versus the other thing. Now, interesting thing about cancer cells is that they lose control of this. So what makes a cancer cell a cancer cell? It's very important. All right? So maybe if you ever take notes, I mean, you should be taking notes. You shouldn't be just sitting there listening to me. But what makes a cancer cell a cancer cell? It's very peculiar. By the way, if you're doing lab 7, you're going to learn about this anyways. And that's why the thing, I would always do as much as I can from the labs because each part is an opportunity to learn. And learning is the point of it. It's not the grade. It's the growth that's important. Whenever you stop improving, there's something that a chance that you've missed at becoming better. That's why I would try to do as much as I can of it. So that part about cancer is super interesting. But anyways, things that make cancer cells cancer cells. So for example, the DNA is heavily mutated. So there's a lot of DNA mutations. All right? There is too much DNA. So it's the idea that the DNA replication is up. So these things. So why does that happen? We'll talk about that in a second. Other characteristics of them. So you see, by the way, the darker parts, like I said in prophase, are typical of the chromosomes, right? So when you look at a cancer cell, and this is an example of them, look how they just look dark. That's because the inside of the cell is primarily filled with this bunch of chromosomes. So you have like, instead of having a cell that has 2 copies of each chromosome, you have 1,000,000 copies of each one. Right? Not 1,000,000, but like hundreds of copies of each chromosome. It's completely out of whack, right? Cancer cells grow uncontrollably. They basically spend less time on interphase, all right? Which means they spend more time actually dividing. Now this is let me rephrase this because I don't want people learning the wrong thing. Division is still quick. In every case, mitosis is like less than an hour. But there's less time between mitosis. Does that make any sense? They just divide very often. All right? So the interphase is shorter, so to speak. So the cells keep going. They tend to live in, they sometimes produce toxins, or at least produce a carbon dioxide rich environment. Why? Because if you're dividing that quick, you're producing a lot of energy, consuming a lot of energy to split that quick. So they're going to be cells which are doing a lot of glycolysis, a lot of cell respiration. So, in fact, we use this to identify them on a PET scan. A PET scan, I'll show you a picture of a PET scan. So this is how I found out that what I had inside of me back in the day was malignant, because I never actually opened myself up to see it. Nobody actually went in there and took a biopsy. But the way that is that the parts that are active light up. So if you give someone a cocktail of radioactive carbon sugar, right? The part that lights up the most will be the cells which are dividing the fastest. So when you have a tumor, by the way, functional MRIs give you a similar picture of activity, but in a different method, okay? But that's used more to see which part of the brain is active when you're doing a certain task. This is more, there's a growth there. Is it malignant? Typically, if there is a cancerous growth, right? So you see, the CT scan will only show you that there's a shape there that shouldn't be there. But the PET scan also shows you that it is highly metabolically active, right? And so that's suspect for a tumor, right? So, and that's kind of how, you see, so the breast cancer spread to the spine, and there was a spark of activity right there that shouldn't be there. That's not a part. So if you look at a regular scan, it's not normally there, right? And so that's a focus. See, this is another one that's pretty cool here. I'm going to show you that one. Where was it? All right, so you see the CT scan only shows the shape of the inside. By the way, CT scans are a series of X-rays done of each slice of your body, and then they put it together to make a picture like that. So they take a slice, a slice, a slice, a slice, a slice, so you can actually get a 3-dimensional picture if you wanted to. Right? And there are 3-dimensional CT scans. And of course, you see that there's a tumor there because there's a growth that shouldn't be there. But not only that, it is highly metabolically active. So this is called a CT-PT scan. It combines both things, and it helps you pinpoint. The machine that did my surgery does a CT scan right before, and then the doctor just goes, marks the part that he wants to burn, presses a button, and then the laser goes and doses it, right? Gamma rays. Each gamma ray is completely inert, but when they all converge, it becomes intense, and it acts like a little razor that not cuts, but literally toasts whatever's in there. And since it's probably not, this is the other thing, because you stop performing the function. Now these will be true of all tumors, benign or malign. Okay, so tumors are dysfunctional, toxin-producing, carbon dioxide increasing, uncontrollable growth, short interface, too much DNA, mutated cells, right? Also, characteristic of them is that they do not obey the no touchy rule, all right, or the touchy rule, I guess, which is when the cells touch, they stop growing because they know there's something next to them. Now, that's how the cells of my hands, which are constantly always dividing because I always screw them up in CrossFit, all right, so they are growing because they know there's a gap there that's not supposed to be there. So they're not touching another cell, they tend to grow. It actually induces growth, and that's how the cell knows to heal the gap that's missing. But when the cells are already touching, that inhibits the growth. Because the cells ignore that signal and just keep going. And what ends up happening, it was in the presentation that I was showing you, is that they actually pile up on top of each other. So cancerous tumors growing in a petri dish would actually, instead of forming just a layer of cells, the cells will pile up and grow outwards. In the body, that becomes a tumor, right? Now, the difference between a benign and malign tumor is that a benign tumor has a tendency to metastasis, right? Which is the idea of metastasize, which is the idea it spreads somewhere else in the body. It also tends to produce toxins that hurt you, or it interferes with function. So it's not about that, if it's just growing there but not growing anywhere else and not producing any toxins or not disrupting the organ, then it's not malignant, right? But if it's taking the space that your liver is supposed to be at, so your liver is becoming something else because the cells are slowly dividing, it suffocates the space of the liver. The cells of the liver stop being liver cells and stop doing what liver cells do, and then now it's malign because it's interfering with the function it's supposed to do. You might as well do a surgery and remove it, or do chemotherapy and destroy it, right? So that's like, you may ask yourself, like, but you blasted your brain? Didn't you lose neurons with that? Well, certainly, but those cells were killing me anyways, so might as well take them out as opposed. And of course, your immune system is supposed to do that, and it recognizes those cells are weird. By the way, that's the other thing that's peculiar about cancer cells. They have broken self-destruct buttons. Right? The self-destruct, which is called apoptosis. Apoptosis is not working. So the cells are supposed to recognize that they're going to be in trouble and say, okay, self-destruct, kill myself, send the lysosomes, destroy the mitochondria, destroy everything else, and then it kills themselves, like literally. So cells have mechanisms to identify themselves as problems. But if that mechanism is also already mutated, of course it's not going to work, right? And then the cell goes towards becoming a cancer cell. Another thing that's interesting about the way cancer works is, okay, so why do these things happen? How does this happen? How does the cancer get to this point? How can we stop it? So the idea is there are genes designed to prevent this from happening, right? So, for example, genes that control the cell cycle. So if you control the cell cycle, you're preventing it from growing uncontrollably, correct? There are genes that are involved with recognition of mutations. Not yet. Is it? Is it 45? I'll pick it up from here next time. We were talking about how does cancer, what's the inception of cancer? So we'll talk about cancer's inception. Actually, next time is Lab 5 Part E. All right? Make sure you do that. We actually may not have another lecture section until after the break, after the test, after I come back. Which means for a while we're going to be working on something else. Guys, in the meantime, those of you who didn't study division, Mendelian genetics, and chromosome inheritance, make sure you read, okay?
Have a great day, Lena.
See you later.
I do not know how to go the world. We are strong enough, we will survive. If we don't, then we're reading.
I'll talk about it later with you, some other homeroom. But I just watched this video that talked about extinction. And then the person at the end talked about